Okay, so we have covered step 1 until step 5. So remember that step 1 until step 5, those are the steps involved in energy investment steps. So now we move on to the second portion, which are the energy releasing steps. So starting with step 6. So now that we have two molecules of G3p, so each one of these, okay, you have two molecules, yes, because this have DHAP has been converted to G3p, so now you have two molecules of G3p. So each G3p will undergo step 6. So what happens is that it will undergo transferring of electron, and the transferring of electron will involve redox reaction. So G3p will undergo oxidation, and oxidation is a process of losing an electron. So G3p loses electron, and this electron that it has lost will be given to an electron carrier, and this electron carrier is called NAD+. Nicotine amide adenine dinucleotide. So it carries electrons. So when it gains the electron, it's basically will be reduced. So NAD plus, as it receives electron, it will be reduced into NADH. And where do you get this H? It's actually, of course, it involves water molecule as well. So this is how you get NADH. The H actually comes from water molecule. Okay, now after you have oxidation and reduction, and then the oxidized G3p is then phosphorylated. So remember, what is phosphorylation? Phosphorylation is the adding of phosphate group, okay, to a substrate. So in this case, we add a phosphate group to the oxidized G3p, glass radiohydrophosphate. Now the question is, I told you that the first until the fifth steps are energy investment, but the moment we start step 6 until step 10, those are energy releasing or energy payback. But why is it? So now we are adding phosphate group. Do we have to use our own ATP again? No. We get this phosphate group not from our own ATP reservoir. We actually get this phosphate group from the phosphate ion itself. Okay, we get this from phosphate ion itself. So we simply take phosphate ion and add it to G3p. So when you add phosphate group to G3p, we are converting it to 1,3-biphosphoglycerate. So after oxidation and reduction, after we oxidize G3p and then we phosphorylate it, and therefore we convert it into 1,3-biphosphoglycerate. So you can see, since you have two molecules of G3p, so this is the first molecule of G3p, this is the second molecule of G3p, and because we add another phosphate group, so this is the initial phosphate group that we added previously, so this is the second phosphate group, and this is the reason why we have 1,3, because one phosphate group is attached to the first carbon, right? And the second phosphate group is attached to the third carbon. Similarly with the other molecule of G3p. The enzyme that catalyzes the oxidation and phosphorylation of G3p is called G3p dehydrogenase, glass radiohydrophosphate dehydrogenase. Okay, before we start, you have to know that 1,3-biphosphoglycerate is a molecule that can easily transfer its phosphate group. Alright, so this is a molecule that have no qualm in removing its phosphate group. So this is what happened because it can easily give up its phosphate group. So 1,3-biphosphoglycerate will undergo hydrolysis. It uses water to lyse phosphate group from itself. So removal of phosphate group. So we remove one phosphate group out, okay, from the 1,3-biphosphoglycerate, and this phosphate group is given to ADP. So the question is, where do we get this ADP? We go back step 1. Step 1, remember in step 1, here, in step 1, we use our own ATP, right? To get the phosphate group, remember? And we convert the ATP to ADP. This is the ADP that we give this phosphate group to in step 7. So we give it to the ADP from our own reservoir, right? And when you react an ADP with a phosphate group, we form ATP. So this is the ATP that we form to pay back the ATP that we use in step 1. Since we have 2 molecules of 1,3-biphosphoglycerate, so this phosphate also be given to another ADP. Remember, if this one is the first step, remember in the third step, we also use our own ATP from our own reservoir. So we give this phosphate group from another molecule of 1,3-biphosphoglycerate. We react it with ADP, therefore forming an ATP. So we invested 2 molecules of ATP. So in step 7, we pay back. Okay, in step 7, we pay back. Okay, so in step 7 involve hydrolysis as well as phosphorylation. So you have to bear in mind this type of phosphorylation is actually quite different from the phosphorylation in here, step 1 and step 3. In step 1 and step 3, this is the type of phosphorylation we call it as oxidative phosphorylation because it involves oxygen as the ultimate electron acceptor. But in step 7, okay, this type of phosphorylation, right? Because we add phosphate to ADP. This is not oxidative phosphorylation. This is what we call as substrate level phosphorylation. So you still add phosphate group to a substrate. However, it basically requires less requirement in order to add phosphate group. The only requirement is that we only need more negative standard free energy of hydrolysis. Okay, the enzyme that catalyzes the hydrolysis of phosphate and the substrate level phosphorylation of ATP here is called phosphoglycerate kinase. See, kinase, same. So by now you should be familiar that kinase is the enzyme that add phosphate to a substrate.